This machine called HPLC is capable of detecting small molecules, metabolites and chiral molecules at nano level. We have used this machine exclusively for analysis of chiral molecules and drug intermediates.
This is an inverted fluorescence microscope from Olympus. It's an I-81 model. It is one of the first fluorescence microscopes that the school acquired.
Thank you DST-FIST for providing us quantitative real-time PCR from Applied Biosystem. This is the recent version, Quant Studio 3. We use it for checking the gene expression and for genetic testing.
The high-speed centrifuge that obtained through the FIST program is useful to protein purification and as well as subcellular fractionations.
We thank DST-FIST for this multi-mode plate reader. This multi-mode reader has allows us to read both at absorbance, fluorescence as well as luminescence.
This machine is called Lyophilizer, also procured from DST-FIST grant. It is mainly used for removing water from biological samples.
The infrastructure created through the funds from the DST-FIST has immensely helped our MSc students, PhD students and as well as faculty. Now our MSc and PhD students have an access to the high-end equipments. We published so far 50 high-end papers and Q1 and Q2 ranking papers like Nature Communications, PNAS and ACS catalysis. That reflects our departmental innovation and research because of the infrastructure developed through the DST-FIST program. So this infrastructure and extramural funds further enhances our research output and innovation, and we hope to achieve much higher goals in the coming years from the funding of DST-FIST.